Hello. So we are wrapping up week 3 with one of the topics in biology that is both super, super cool and also really kind of notoriously complicated. I'm here with Nano. He is joining me at my desk right now. He's feeling a little extra spicy tonight. We introduced him to the outdoors of our porch, and he's kind of being a little bad about it. So he might be a little bit cranky in our video, but that's okay. Cats are biology, I guess. That's maybe my argument. So just a couple of quick notes before we get into it. So your discussion board, if you haven't already posted that, please get that in by today. That is Thursday. I'm filming this on Wednesday night. And go ahead and get started on your sustainability topic outlines. So some of you had let me know that there was trouble with the page when it was initially posted. So if you had been putting it off because you were waiting for that to get resolved, it is now resolved. So go ahead and take a look at the assignment description. Essentially, I want you to zero in on one component of your chosen sustainability topic and make a detailed outline for it as if you were going to be writing an essay about that topic. And then finally, if you could do me a favor and take the poll that I'll be sending out about biology conference periods. We've had sort of mixed attendance with the different ones, so I want to, one, figure out, if there are other times that would work better for people, and two, just figure out if certain times that are not as well attended are because they don't work for people, or rather just that they're not needed. Right now we have 4 conference periods a week, but I'm considering trimming that down or at least changing them. So your response to that is super helpful to me, and it shouldn't take very long at all. It's just a couple of yes/no questions. Okay, so here's Nano. Seems like he's strapping in for a second. Maybe he'll hang out. We are going to talk about cellular respiration. So I sort of left you on this note last time of thinking about endosymbiotic theory. And this is the idea that the powerhouses of cells, so the chloroplasts and mitochondria, are actually the descendants of ancient OG prokaryotes, right? And that's super cool and really weird, and makes a lot of sense. Really, the evidence is really stacked in favor of that, and it's very, very cool. But so we didn't talk yet about the actual process of cell respiration and how these really incredible organelles are creating energy, how they're creating ATP, those energy dollars of the cell. And so the way it does this, of course, is the process of cellular respiration, which we've mentioned before and is what's pictured here. And the process of cell respiration is really divided into 3 stages. So we've got the initial breakdown of that stored chemical energy, glucose. We've got the process of glycolysis, where we're breaking down glucose. We can see it right in the name. Glyco is a prefix that indicates it has to do with sugars, and lysis, as we've learned in previous terms, is a suffix which means to break. So breaking down of sugar. Following that, we have a process called the oxidation of pyruvate, or pyruvate grooming, followed by the citric acid cycle, also called the Krebs cycle. And then we have a very neat sounding process at the end called oxidative phosphorylation. And so all of these are kind of scary sounding and definitely a little, trickier. I would say this is another one of those situations where we're getting into some metabolic pathways. And so we're getting into some molecular biology concepts and processes here. So take time with it. Know that this is one of those notoriously difficult topics. And so if you're struggling with it, it's totally normal. and appropriate, really. If you're struggling, it just means you're thinking hard about it. But if you do need help as we're going through, please come see me. Shoot me a quick email, post something on the Q&A bulletin board, or set up a time for us to video chat and we can quickly go over that, or hop into a conference period, whatever winds up making sense. So, okay, let's get into it. So glycolysis. So again, this is the process where we break down, we lyse sugar, glucose specifically. And so glucose is a really interesting molecule because it's made up of 6 carbons. And so 6 carbons linked together. This is a carbohydrate, which we learned a little bit about a couple classes ago. And so the inputs for glycolysis is a molecule of glucose, 2 molecules of ATP, that again is adenosine triphosphate, so an adenine analog plus 3 phosphate groups attached. And then the last input is going to be 2 molecules of NAD+. So we have seen something that looks really similar to this before in photosynthesis. So we saw NADP+. So the addition of the P chemically, molecularly really matters. When we start changing around the ingredients of a molecule, even slightly, they change quite a bit, and their behavior therefore changes quite a bit. And so this process is going to occur in the cytoplasm of a cell. And again, we are breaking down glucose, but the end product of that breakdown is going to be 2 molecules of something we call pyruvate or pyruvic acid. You may also see it called. For our purposes, pyruvate and pyruvic acid are the same thing. So if you begin looking at other resources, I want you to be able to recognize both. There is a slight difference, but that's not something that we'll cover in the scope of this class. But so we break down glucose into 2 molecules of pyruvate, and pyruvate fundamentally is a 3-carbon molecule. So we take a 6-carbon molecule, and then we essentially are splitting it in half, and we're creating 2 3-carbon molecules. And then the other output of this, which is pretty interesting, is that we wind up with 4 molecules of ATP plus 2 molecules of NADH. And so if we remember from photosynthesis that our NADP plus was the oxidized form of NADPH, which went on to the electron transport chain to facilitate those chemical reactions in the Calvin cycle, the dark reactions of photosynthesis, we may also, kind of project that knowledge onto this. They do function really similarly. So what we have really is an input of glucose, an input of energy, and then an input of an electron carrier. That's the NAD+. And then at the end, we have let me rephrase that. We have the input of an uncharged electron carrier. That is the NAD+. And the output then is 2 molecules of pyruvate, 4 molecules of energy, that ATP. So we've doubled our energy output from our input. And then we also end glycolysis with 2 molecules of a charged electron transporter. That's really what NADH's job is. It's to move electrons around from metabolic process to metabolic process and to facilitate different parts of the chemical reaction, as we'll see soon. And so we call this process anaerobic respiration. When we say something is aerobic, what we're saying is that it requires an input of oxygen. And so to say that something is anaerobic means that it functions in the absence of oxygen. So this process is occurring irrespective of an input of oxygen to the cells, which becomes important as we go through this. And then finally, for this slide, what I want to point out is that this chemical reaction here really summarizes the process of glycolysis. And this is something that you should know and feel comfortable with. So again, this C6H12O6 is the chemical formula for glucose. So we're saying glucose plus 2 molecules of ATP plus 2 molecules of NAD plus yields 2 molecules of pyruvate and 4 molecules of ATP and 2 molecules of NADH. So this is kind of the process. So we start up here. I made this diagram myself. I'm very proud of it. We start up here with our glucose. And then what's happening here is we undergo a reaction where we input our 2 molecules of ATP. And then as we input those, what's happening is one of those P's, one of those phosphates, remember that Pi stands for phosphate ion, one of those phosphates from each ATP molecule that has been input is given up and sacrificed and sort of stuck on the end of that glucose molecule. And so, which I'm just now realizing I've drawn with 5 carbons. Each of these purple circles are representing a carbon. This is a 6 carbon molecule, not a 5. Sorry about that. Let's pretend this is 6. And so we input 2 ATP molecules. Each ATP gives up one of its phosphates. So then each of those ATPs becomes a DP. Again, that's adenosine diphosphate. So 2 phosphates attached to that adenine analog. And so we attach those 2 phosphates that were given up to our molecule here. And what this does is this creates a molecule that is very, very unstable. This form is not desirable. This form is electronically, unstable. I guess I'm struggling to come up with another word for that. But so the point of that being that it really, really wants to split apart at that point. It no longer feels comfortable remaining as a 6 carbon molecule. And so what happens is the addition of these phosphate groups changes the energy such that the molecule has no choice but to split in half into 2 3-carbon molecules. And this is much, much more stable. And so at the end of each of these, right, we slap on a little hydrogen. That becomes important in just a moment, but we've still got these phosphate groups. And you may notice if you're skipping ahead, this is our pyruvate molecule here, our pyruvic acid. It doesn't have any phosphate groups attached to it, but these guys do. So this is what we'd call an intermediate to the chemical reaction, as is this. We'd say this is an intermediate also. And these have specific names, but they're fairly long and they're fairly involved. And I don't know that it helps you much at this point to know their names, but if you are interested, certainly go look it up and learn them. But so we're here and we've got these more stable 3 carbon molecules with a phosphate group attached to one end and a hydrogen group slapped on the other end. So, but we're not done because again, we need to get down to pyruvate, which does not have a phosphate group attached to it. So we bring in our electron absorber. We bring in NAD+, and her job is to scoop up that hydrogen atom here and become NADH. Her job as NAD+ is to become reduced. We're going to reduce that overall charge by picking up one of the electrons. And so this is going to happen to each molecule. And then further, because we need to get rid of the phosphate, well, ADP is going to come back in and create ADP is going to come back in and pull off the phosphate group from each of these 3 carbon molecules, regenerating ATP in the process. And so then this process occurs twice. And so what we wind up doing is we wind up having our initial input of ATP regenerated, and then we build 2 more on top of it. And so at the end, we've got our 4 ATP molecules and our 2 molecules of pyruvate. So pyruvate ultimately, sort of, is going to move on to the citric acid cycle, but it can't go as is. So firstly, the pyruvate or pyruvic acid is going to be transported to the inside of the mitochondria. And so it's going to do this through a transport protein. So this is just a big protein molecule stuck into the plasma membrane of the mitochondria. And this is a doorway by which this large molecule, this 3 carbon molecule, can enter. And so it's going to hop into the mitochondria. And when it does, it's going to undergo a couple of chemical reactions itself. So we call these chemical reactions the grooming of pyruvate, and it's sort of like getting a fancy haircut to be ready to go into the citric acid cycle ball. And so what's going to happen is if we look at the molecular structure here, we look at this primary structure of the pyruvate molecule, we'll notice that we've got our 3 carbons here, and at the end, one of those carbons is involved with a carboxyl group, which we've also seen in our amino acid structures. And so pyruvate's going to enter in through that transport protein, and the first thing that's going to happen is that carboxyl group is going to pop off, because we need to put something else in its place. So it's going to pop off. We're just going to cut that length of carboxyl right off the top. But wouldn't you know it, if we look at the ingredients of a carboxyl group, we've got 1 carbon, 2 oxygen. That's CO2. Carbon dioxide, same exact thing. And so the byproduct of the pyruvate oxidation, of this grooming of pyruvate, is carbon dioxide. This is one reason that when we exhale, when we respirate, we are eliminating CO2, carbon dioxide, as a waste product. And so now that we've lopped off this carboxyl group and we've lost it in the form of CO2, the next step is that our electron shuttler, NAD+, is going to come back in, and she is going to scoop up the excess hydrogen from pyruvate after it's lost to the CO2, and it's also going to scoop up a excess hydrogen from this molecule here, coenzyme A, also abbreviated as CoA, written like this. And so NAD+ comes in, soaks up a hydrogen from pyruvate, becomes reduced to form NADH, and then it's also facilitating the release of a hydrogen atom from this coenzyme A, and so that's where this extra hydrogen atom is coming from. So these guys move off to the side. They're going to go and do something else a little bit later. Coenzyme A then is going to essentially take the place of the CO2 that we lost. So it's going to take the place of that carboxyl group on the pyruvate. It's going to be bound to that second carbon right where the 3rd carbon was. And so this S we're seeing here stands for sulfur. And so there is a sulfur group involved with this enzyme. And so when this coenzyme A hat gets stuck onto the head of our groomed pyruvate, what we wind up with is a molecule called acetyl CoA, which is what is going to ultimately enter the citric acid cycle. And so if this is sounding a little bit familiar, CoA is actually a type of B vitamin. And so if you've ever heard or seen, maybe you've got B vitamins at home, if you've ever heard or seen this before, you may have read that B vitamins are sometimes thought of as useful for increasing energy levels, or you may see, I know like on my vitamins, it says something like helps your cells create energy. This is the basis by which they're able to make that claim. So there's certainly some debate about, you know, the efficacy of vitamins and, you know, like maybe they work pretty well, maybe they don't, maybe certain ones work better than others, maybe certain types work better than others. That's something that, you know, we still need a lot more evidence on to sort of decide either way. But this is how they're able, this is the basis by which they try to make that claim, which I think is pretty interesting. Okay, so where we're going to leave off is we are going to very gently look at the citric acid cycle, also called the Krebs cycle. So this was named after a German scientist who discovered it, though he was not the first one to discover it. More on that later. But in general, what I want to, you know, sort of leave you with for today is just this sort of broad stroke approach to what happens in the citric acid cycle, or the Krebs cycle. So acetyl CoA is going to enter, and remember, acetyl CoA is that modified pyruvate that was just created in the last step. So this is all occurring in the mitochondria also. So acetyl CoA enters this cycle. It's been made. It's now entering this cycle. And then essentially we have a bunch of reactions occurring. We have a lot of enzymes doing good work. We have inputs of we have movement of electron carriers, our NAD+. We have release of CO2, and then ultimately at the end of our process here, what we've done is we've created a bunch of molecules that are going to go off to another process occurring in the mitochondria. They're going to move off to an electron transport system. So if your memory is a little bit fuzzy already on the electron transport chain mechanisms from photosynthesis, I recommend strongly that you go back and just kind of review those pieces before we go further. So before we finish this up on a Tuesday of next week, because they're remarkably similar. And if you felt fairly comfortable with the mechanism of photosynthesis, you'll be in a lot better shape going into the electron transport system of cellular respiration within animal cells. And if you're not feeling comfortable with that, now is the time to come see me and get feeling comfy. It's fascinating stuff. It's really cool. It's really kind of tricky though to get the hang of. And this looks fairly, you know, nice and simple for right now, but she will not stay that way, unfortunately. So with that, that's all I've got for today. Nano says hello. He's kind of off to the side. Yep, that's just mostly his bottom. Oh, here he is. Nano says hello. I say hello. Would love to see your faces. Let me know if you have questions, and I will talk to you soon.